Well, I'm happy to be introducing today. Oh, silence. That's what I like. I have a few announcements before I do introduce. Number one, at the end of the show today, we'll be taking questions, and I would remind you to turn on your microphones so that people who are zoomed in can also hear your questions. We will be taking questions by Zoom through the little chat box, and so please don't hesitate to ask questions, everybody here as well as in the outside world. I have a couple of announcements, and that is to remind you about our research day, August 9th. Please register, and also please register for our CBR 20th anniversary extravaganza on July 7th. Next week, we will have Karen Chung's lab speaking on our Wednesday seminar, but this Wednesday, we are delighted to introduce Leonard Foster's laboratory will be talking to us, and he will be taking over from me to introduce his laboratory as soon as I shut up. Leonard Foster is professor and department head of biochem and molecular biology. He started out at SFU as an undergrad and then saw the light and went to school where I went to school at U of T to do his PhD. And I am now going to shut up. So, Leonard, thank you very much.
All right, thanks very much for the introduction, Ed, and thanks to the other Ed and Mira and Parvin for keeping CBR on track and arranging these seminars, and for the funding that I know many of our labs receive through the CBR. So that's obviously very much appreciated. I'm not going to do most of the talking today. We're going to hear from two people that work in our group, and they'll do most of the talking, hopefully, depending on how time goes. I will fill in with a little bit more. And then, like Ed said, we'll take questions at the end. So first up, we've got Armando Alcazar Magana, and he will be, so actually I should back off. Armando is Mexican originally and did his schooling there, and then has been, or up until about a year and a half ago or so, was doing a postdoc in Oregon on metabolomics and many different aspects of metabolomics. And about the same time, I am in sort of my own capacity as a researcher, but also through discussions with the Life Science Institute here, we're really keen to build some metabolomics capacity at UBC. And this is not to take away from the others who are already doing metabolomics at UBC and know more about it than I do, but there was some motivation to try to develop a central facility that would be able to offer services in metabolomics in the same way that my own group offers services in proteomics. And so we were unbelievably lucky to recruit Armando here. I run out of superlatives to describe exactly how far he's taken metabolomics at UBC since he's been here for about a year and a half. The other thing you should know about Armando is that he likes hiking possibly more than I do. So he's a very good fit for the lab in that way. And I think you'll see a little bit about that today. So Armando?
Thank you. Can you hear me here? Thank you for the introduction, and thank you everyone for coming. Since I arrived here, I feel like I'm at home, even so far from my home, but I feel very welcome in the group. And as Leonard said, one of the things that I really enjoy here is how beautiful is Canada and to hike. So, sorry, go back. I want to show what we have in the facility. We are very lucky that we have very fancy toys, state-of-the-art instruments. We have a timsTOF for high-resolution mass spectrometry. We have an impact tool. We have also a TOF from Agilent, and all our instruments have a UHPLC in the front, and so we can basically do separation and then the measurement or detection of the molecules in the instruments. So we live in the omics era. So basically, we start from the genomics, then we have the transcriptomics, proteomics, and metabolomics, and the regulatory mechanism is increasing, and also the complexity is increasing. Basically, for the genome, we start with 4 bases, right? That basically in modification of those, and basically we start then the transcription, and then we have 20 amino acids that they can do proteins, and we know it's a lot more complicated than 20 compounds. But when we go to molecules living in organism, any living organism, basically we can have more than 100,000 compounds, and all of those with different chemistries, and we have different ways to detect them. And so that is basically what we do with metabolomics. So to do genomics and transcriptomics, we basically use sequencing. And for proteomics and metabolomics, we do mass spectrometry. So that is the thing that gave us together this. In the Foster lab, we have instruments that we use for the proteomics and instruments for metabolomics that basically they are the same. Basically, we just need to change some parameters. And so basically the study of metabolites is like a snapshot of what is happening right now in an organism, in a tissue, in a cell. And by that, we can get an idea of how is the metabolism, the energy production, signaling pathways, and overall, we can see how is the health of that cell, of that system, that tissue. So to identify the compounds, we basically have our sample, we extract the samples, and then we have a extraction protocol, and then we introduce to an instrument, the mass spec. And we have data in many dimensions. One of them, for example, is retention time. If we have a column, a separation, we have the mass charge, and we also have the intensity. So that is 3 dimensions. But in addition to that, each one of these dots that you see here it has fragments. Basically, the instrument can take a molecule and make fragments, and then we have an idea of the structural composition of that. And we also can have the area under the curve for each one of these compounds and have the quantification information to compare among samples, treatments, whatever you can name it, the treatment. So basically, with this technique, we can have the precursor mass, and with that, we can get the elemental composition. This is especially true for small molecules. When we have big molecules like proteins, this is not that simple. We need to, we cannot get the elemental composition that easy. We also have the isotopic distribution, and that is also important information because when we have for example, a sulfur atom, that is, we have 2 most abundant ions, like sulfur 32 and 34, and then the isotopic pattern is different. All that information, we can include it to get the annotation, and also, as I mentioned, the fragmentation. We acquired all that information for a library that we call the primary metabolism library. That is about 600 compounds that you're very likely to find in mammalian systems or in almost any kind of living organism, the most common ones. So we are offering that service. In addition to that, we have another library of natural products of compounds that you will find probably in wine, in beer, in some antibiotics, and basically in plants, and also a sponge or some ocean organism. And so with those databases, we can have different confidence in the annotation of the compound that we see in an organism. And basically, we have a database that match against the new ion that we see from an unknown sample. And if we have this matching, then we can annotate with different levels of confidence. If we have experimental information and we know that we run an authentic certified standard under the same conditions, we can call it tier 1 or level 1 annotation. Otherwise, it could be level 2, level 3. There are 5 levels. And in addition to that, we have access to some databases like Medlin, NIST. We have the Human Metabolomics Database, Snapsack, KEGG, and there are many others, the Metabolomics Association of North America, etc. So we can do this targeted metabolomics that basically we try to see as many as possible. We can see many thousands of compounds in plasma, in urine, saliva, biofluids, and basically any kind of, we are focusing in clinical and biological samples. Even though the instruments can be also used for environmental application, now we are focused in this. And the clinical samples, for example, could be persons with different degrees of progression of disease, for example, diabetes, of cancer. We can have groups in that way, but also if we study like some other organisms, we can compare control against mutant. And not only that, but also treatments of those organisms. For example, a model of mice that we can feed different drugs or different food, or even with or without microbiota, to see what the microbiota do with those compounds. A lot of medicines, the effect of us is the metabolize the microbiota do with that. And so if different person has different composition, it could be different, different response of the treatment. This is basically the way that we do our workflow. We have the data collection, we have feature detection, statistical analysis, and feature annotation. We always try to do randomized samples, so we don't have a batch effect, and we only capture the features that are really, really important for us. And the slow part in this analysis is, well, running the samples is about 20 minutes per sample. And then the feature detection, we have many parameters to tune according to the sample. And the part that we are suffering is the data processing time. That is the slow part, because basically we need to see every molecule. There is a lot of things happening, like in source fragmentation, we have isomer, we have a lot of false positive that basically we need to check. But now with machine learning, we are trying to move that forward. Another analysis that we are doing, actually we already did samples here for the Life Science Institute, is in the lipidomics analysis. And here we can see again, the intensity, we have the retention time. And the good thing of lipids is that we know the retention time about where they show up. And some of them show up in negative ion mode, like fatty acids. Some of them show in positive mode. And basically, this helps us a lot, because if we see, for example, a triglyceride in this region, we can remove it. That is still manually, but we know we can remove a lot of false positives in lipidomics. So it's a pretty mature technique. This is another type of comparison that we do in the past. This is a group of people, healthy people, controlled, and the other group is people with different, with 3 arteries, with coronary artery disease. And so this plot has a lot of information. I can show a little bit. We have the retention time again. We have the mass chart, and you see it's like a mirror, right? So below is the healthy people, and above is the people with coronary artery disease. And we can see, like, for example, triglycerides and ceramides are much, much higher in this person. But many of the phospholipids are lower. And all these features that you see here is only statistical significant features. And the size of the sphere is the fold change. And also the darkness is the p-value. Okay. And another thing that we do is to do deep characterization of some samples. For example, we are working in the composition of honey, and we do the extraction of the compound, removing the sugar, basically. And all these ions that you see here is already after removing the noise of the experiment. And each one of these ions basically has fragments, or only fragmented. And also we have the collisional cross-section, like it's basically the surface of the molecule when we blow it against a current of gas. So we are collecting this data, but this can be applied for any kind of tissue, any kind of cells or plants, wine, whatever. It's like a fingerprint. And one thing that we are very interested in working now is what we call the molecular networking. And I want to show a little bit why. Can anyone say what is this object and this one? Any idea? Okay. They're still there. So what could be this object? It's a person, right? And we know because Leonard is here, and it was a bear or some, I don't know, a danger. He will not be that close, maybe. And the other we see is a rock because we saw other rocks. So also, if we didn't know that there were more persons here, probably we will not know that it's a person. So molecules basically are arranged in organisms that way. So by knowing the neighbor compounds, then we can be sure of a class at least, or the identity of some. So when we have this extraction, and then we have all the features, thousands and thousands, but they are disorganized. So basically, we don't know how, if they are connected in pathways, or if a pathway is upregulated or downregulated. But now we have some tools to try to organize molecules that are similar in the fragments. So whatever is sharing fragmentation, about 70% or more, they are connected here. So if you think how important is this, if we remove that interaction between molecules, we are basically our bodies is made of just dead compounds, right? If atoms, molecules, we separate them. They don't have a consciousness. Probably, this coordination or orchestration of molecules is what makes us feel, stress or fear, or when you enjoy that walk in the forest or in the mountains, right? That orchestration is what it makes interesting, or we can define as alive. So almost finishing. So also we are trying to go to smaller molecules, and hopefully one day we can go down until a single cell. We are working now with a small oocyte from a honeybee, but it is still huge in comparison with other cells, but it is teaching us how to improve our sample prep and all the steps that we need to move forward to work with smaller cells. And this is also a network analysis. And basically, we can have, for example, 2 treatments, and we can represent in the colors. And you see some of them are bigger. Those compounds are more abundant. And also you see the thickness of the edge is different. So that is, the minimum one is 70% matching, MSMS matching. But if it's thicker, go up to almost 1. And so another application of this is that when we analyze, for example, this is a extract of a plant that is called Centella asiatica. And when we have compounds that are very close together, it's very likely that it's an enzyme just moving from one to the other. This is the case of camphorol and quercetin, that basically this is a flavonoid monooxygenase that is just putting a hydroxy group. But probably there is many enzymes in other nodes that are close together. And yeah, finally, I want to say thank you for the funding at CBR, and also Foster Lab is really a fantastic place to work.
All right, thanks very much, Armando. Like I said, we'll save questions until the end. So next up, we have Alessia Palumbo, and Alessia is an honor student in the biochem program here at UBC. They approached me, I think, last fall about doing an honors thesis with our group in this coming fall. And I think it's fair to say more than any other undergrad student I've ever had approach me, they really had their own ideas about what project they wanted to study. And so that's what we're going to hear about today. This is just getting started. Alessia has funding from CBR for working this summer. And then, like I said, we'll be continuing with us in the fall. But it has been a bit of a journey already, getting to the point where we're at with developing the ideas behind this project. And so I'll ask Alessia to come up and tell us about it.
Hello, everyone. As we begin today, I do want to give a land acknowledgement. We are on the unceded territories of the Musqueam, Squamish, and Tsleil-Waututh peoples. Unceded is a word that means stolen, but more than that, it is a word that means that even by the rules of the colonizers, the land that we are occupying is not supposed to be occupied. It's very important to acknowledge the land that we're on and where we are doing our research, especially when doing research like mine, which affects marginalized communities. Recognizing intersectionality and the impact that our research is going to have on other people around us is incredibly important. So to begin, my name is Alessia Palumbo. I use they, them pronouns, and I am, as Leonard mentioned, an honors student in the Foster Lab. I am doing my research on the cellular effects of gender-affirming hormone therapy. So safe and accessible gender-affirming hormone therapy is an important part of working toward equity in healthcare for transgender people and ensuring safe social and medical transitions. However, there is currently a critical lack of research on the health effects of gender-affirming hormone therapy, making it challenging for transgender patients and their healthcare providers to make informed decisions about their treatment plans. This research project is designed to fill the need for further research into gender-affirming hormone therapy to determine the effects that it has on a cellular level. So just as we're beginning, I wanted to give a couple of definitions and a brief chat about what gender is. So gender identity exists in our heads. This is the Genderbread person. It is a very good resource if you want to check it out, and it gives a pretty good explanation for all ages about what gender identity, expression, sex, and sexuality are. And then I just wanted to go through some terms as well. So first and foremost, some terms that you will be hearing a lot throughout my presentation. AMAB, which stands for assigned male at birth. This is the preferred terminology for somebody who, when they were born, was given the male designation, and that was written on their birth certificate. In a similar vein, AFAB stands for assigned female at birth. And then I also wanted to give the definition of transgender woman or transfeminine, which is just somebody who identifies as a woman or on the feminine spectrum of gender, but was assigned male at birth. In the similar vein, transgender man or transmasculine is somebody who identifies as a man or on the masculine spectrum of gender who was assigned female at birth. And then up next, the last term I have on the slide is gender-affirming hormone therapy, or GAHT, which we will be talking about in the next slide. So gender-affirming hormone therapy is the use of hormones, anti-androgens, anti-estrogens, and other hormonal treatments with the aim of increasing congruence between one's gender identity and their physical body. This allows predominantly trans and non-binary folks to align their secondary sex characteristics with their transition goals. And while the goals of gender-affirming hormone therapy are incredibly individual, sometimes the treatments haven't gotten to that level of individuality, so there's a lot of common treatment plans that are used for just about everyone. So this research project is going to focus on the hormones used in feminizing hormone therapy. So feminizing hormone therapy is the use of hormone therapy to change secondary sex characteristics from masculine to more feminine or androgynous to align with a person's gender identity and transition goals. This is frequently use of anti-androgens to suppress gonadal production of testosterone, and as well as the use of estrogen or estradiol predominantly and progesterone to induce changes in secondary sex characteristics. Very briefly, estradiol is an estrogen steroid hormone and the major female sex hormone. It is known best for regulating the menstrual cycle, but contributes to regulation in many different cell tissues in the body. The secondary sex characteristics that estradiol is associated with is breast development, soft and less oily skin and hair, fat distribution, and differences in bone maturation. Progesterone is a progestogen sex hormone. It is also best known for the regulation of the menstrual cycle and during pregnancy. But the secondary sex characteristics it contributes to is once again breast development, fat distribution, and changes in body hair growth that occur during puberty. In AMAB people, if we remember, that stands for assigned male at birth. Progesterone in lower amounts allows for sperm development and is necessary for the production of testosterone. So a typical treatment program for a trans feminine person who is undergoing gender-affirming hormone therapy would look like the use of estradiol in combination with an anti-androgen to stop the gonadal production of testosterone, but not the cellular production of testosterone because it still is a necessary hormone. The use of progesterone is very dependent on whether the patient and the doctor agree that it would be a good fit for them, or if the transgender person themselves has done the research and wants to try it out. So as I mentioned earlier, I'm going to be using cardiomyocytes and B lymphocytes. Oh, I don't think I mentioned this yet. As I'm mentioning now, the cells I will be using are cardiomyocytes and B lymphocytes. So here's a little bit about why I decided to go for cardiomyocytes. Basically, cardiomyocytes in people who are on gender-affirming hormone therapy, specifically feminizing hormone therapy for long periods of time, tend to have increased rates of heart problems, and this includes heart disease and heart attacks. So there is current debate about the true effects of estradiol and progesterone on heart health in transgender women. Some believe that progesterone improves endothelial function, and because of that, it may prevent cardiovascular disease. And that there's no sufficient data showing negative effects of progesterone. But other people argue that since synthetic progestins are known to have a negative effect on heart health in cisgender women, They might have a negative, like native progesterone might also have a negative effect on heart health. Regardless, it is clear that more research is needed in this area, especially since AMAB and AFAB people have different relative amounts of estradiol receptors and different types as well in their heart. So next, we are also going to be working with B lymphocytes. So to start with, we know that sex hormones affect how the immune system functions and the activity level of the immune systems. But how is this going to affect the cells at an individual level? And what is happening when these cells are treated with hormones that is typically associated with a different sex? There's little current research on how this affects transgender people, but there's actually a lot of anecdotal evidence that something is changing. For the B lymphocytes, we will specifically be working with 2 cell lines derived from a brother and a sister, which will allow us to better analyze any potential changes that are a result of sex differences, since the cell lines are derived from genetically similar sources. So here we're going to be talking a little bit about what we know, how estradiol and progesterone affect cardiomyocytes. It's important to keep in mind that this research is done predominantly on cisgender people, and it doesn't really take into account how transgender people and people undergoing gender-affirming hormone therapy will have completely different systems. However, some of it might be applicable, and it indicates that there will likely be some changes. So for estradiol, we know that they contribute to the differences, estradiol contributes to differences in regulation and in cisgender women cardioprotection. So estradiol receptors are capable of directly affecting regulation of signaling pathways in cardiovascular cells and also of gene regulation in these cells. They also mediate the protective effect of estradiol on heart cells that we see in cisgender women. And estradiol receptors are found in different amounts across different sexes. They're also found in different varieties and ranges. Up next, we have progesterone. So progesterone, we know to impact cardiovascular tissue in sex-differentiated ways as well. So first, we know that it regulates cardiac repolarization, which is really interesting. And we don't really know what the differences in sex, what differences sex has on this. But we do know that it plays an important role in the development of different sex differences in heart tissue. So P4 also is known to regulate differently than estradiol does, and as shown by how it regulates the production of a lipase in the heart cells, but so it downregulates this lipase, whereas estradiol upregulates it. So up next, we also have what we know about estradiol and progesterone effects on B lymphocytes. This is once again not a lot, and once again studied in predominantly cis systems, which is a little bit applicable, but there are going to be many differences in the research that I'm doing. So estradiol is known for changing the threshold for apoptosis and for activation. This is both the activation of the innate and adaptive immune system, and then progesterone is known for being immunosuppressive, and it is suspected to mediate a lot of the sex-specific differences in lymphocyte activity. So this is both B and T lymphocytes, though we're not quite too sure why or how. And it also regulates antibody production in AFAB people. So sex differences in the immune system in general, there are quite a few. AFAB people tend to have a stronger immune response to infection, but also a higher rate of autoimmune disorders compared to AMAB people. And the genes in B cells that are differentially expressed between sexes are usually upregulated in AFAB people. B lymphocytes contain a lot of estradiol receptors, and estradiol in AFAB people is known to increase the rate of antibody class switching as well. So I just wanted to go briefly through a basic experimental plan and what I'm looking at so far and what we're looking to do in the future. It's important to recognize that I'm doing this research because we don't know what gender-affirming hormone therapy does on a cellular level. And proteomics is a great approach to determining key changes. So what I plan on doing is treating the cell lines with different concentrations of estradiol and progesterone and physiologically relevant levels. So these levels and concentrations are derived from median blood concentrations of estradiol and progesterone that are found both in cis women during different points in their cycle, and also in transgender women who are undergoing gender-affirming hormone therapy. so that it's replicating these levels that you will see in everybody else. So the cells will then be lysed and intercellular proteins will be processed for mass spectrometry analysis. And this proteomics approach will be used to determine which proteins are differentially regulated as a result of the hormone treatment. Small molecules and metabolites will also be collected just to take a look into the metabolomic landscape of how these hormone treated cells are changing. Sex hormones are known to contribute to different energy production, and estradiol has a major role in the regulation of both glycolysis and the electron transport chain. So very quickly, I'm just going to give a quick overview of what I talked about today. So first and foremost, gender-affirming hormone therapy is the use of sex hormones, anti-androgens, or anti-estrogens to increase congruence between one's gender identity and physical body. This is a very under-researched area of science, and we have a lot of work that we can be doing, and it's really exciting to start research on this. So we will be using estradiol and progesterone as which are the most common hormones used in feminizing hormone therapy to treat different types of cells, including cardiomyocytes and B lymphocytes, and see what effect that these hormones have on a cellular level. And finally, we will be using mass spectrometry to analyze the results in both proteomics and metabolomics lens. So I wanted to give a thank you to my lab, specifically Queenie, who is not here today but has been training me, and to Leonard, who let me bring this project forward and start. Thank you. Thank you to the CBR for funding me this summer, and to our future collaborators, which is really exciting.
All right, thanks very much, Alessia. I just wanted to say a couple more words about Alessia's project. So when they first came to me, I didn't know. I mean, I know generally what transgender people go through, like in their transition, but I naively assumed that we knew a little bit more about the effects and side effects of gender-affirming hormone therapy. And so when Alessia came to me, it sounded very interesting. And so we start reading a bit of literature and finding out what's known. And it's unbelievable how much is not known about this area. So yeah, so I think at this basic level, which you could, or basic like cellular level, you could argue, well, it's not really the physiological whole human, but there's so much, just an unbelievable amount for us to still learn. And the future, I think, also looks very bright in terms of moving past cells. So one of the collaborators, Alessia mentioned, Zach Laxman at St. Paul's, is looking at organoids and has different questions, but than us, rather than gender-affirming hormone therapy. But the questions we want to ask will fit very well with that model, and then eventually getting access to clinical samples as well to try to understand better what people who are going through this one particular transition are experiencing. And then, of course, there's the whole the opposing transition as well, that is a whole, going to have to be another project. Anyway, I wanted to pause there and pick up where Alessia left off on the land acknowledgement very quickly. So I was at a meeting a couple weeks ago in Regina, where it was a proteomics meeting, and they kicked off the meeting with a blessing from an elder from the local Métis Nation. And then I had the fortune or misfortune of following right after this person, and I had a land acknowledgement prepared. But her, like, she just brought the room down, or the building down, with this blessing. She went on for 20 minutes or so, and then I stood up and felt fairly foolish. But it made me reflect a lot more on the land acknowledgements, and we're getting pretty familiar listening to them. But I think I would challenge everybody to really think, not just when you hear it, think about the meaning, but think about how we move past that. And we hear about decolonialization. I don't really know what that means. I don't think anybody's got a great definition of what it means. But we have maybe a bit of a better understanding of what reconciliation should be. And these land acknowledgements are pushing us towards that, or suggesting that we need to move towards that. But getting there is the hardest part. Saying and listening to the land acknowledgements is the easy part. But we have to keep moving it forward, or our whole society will kind of get stuck, I think. So anyway, you've heard about a couple of the projects in my lab. Armando covered maybe not a specific project, but rather a group of methods that we are now trying to apply to many different projects, and that we hope others will be able to access as well. That has been kind of one of the defining features of my time here at UBC, is making mass spec-based omics methods available to others. And through that, we also obviously have our own projects going on in the lab. And we are a technology-focused lab, developing and applying mass spec-based methods. And it's obviously been mostly proteomics, but we're moving into metabolomics as well. But we have had our biological focus can be loosely defined under this title, host pathogen interactions. And I'm going to just mention a couple things very briefly. There's way too many projects to cover all here. But we have done a lot of work on protein interaction networks. I'm not going to talk any more about that. We've done some work on antigen identification and how that can be related to vaccine development, and then also understanding what might make a universal vaccine that is useful across the whole world. Surprisingly, most vaccines are not. And then we, maybe a little bit more closely related to blood, we've also been involved in a lot of biomarker projects, but again, I won't have time to talk about all those things. And then the other thing that we're known fairly well for is our work on honeybees, and I will start off with one short story about that. Also trying to keep within the time that we've been given here. So honeybees, again, I know this is a blood research center, but actually we have received some funding for some of our bee work, and I know it is of interest. So just very quickly, the main function that bees provide for humans is not the honey, but the pollination services. And these are 5 of the biggest, most important crops in BC. And our goal in applying proteomics technologies to understand bees is also focused on host pathogen interactions. There's many different host pests and pathogens of bees, and viruses are kind of the most important one, as you might argue they are for humans as well. But the main vector of most of the viruses in bees is a little mite called Varroa destructor. It's a parasitic mite, and it functions very similar to how mosquitoes do for humans in vectoring viruses. And so while the viruses are problematic, there's no adaptive immune system in bees. And so there's no real antiviral defense apart from some of the siRNA mechanisms. And so really the secret is to go after the mites. And turns out that there's a behavior in bees that makes them more resistant to mites. It's called hygienic behavior. We're measuring it here by ladling liquid nitrogen onto these frames of bee comb. And then you put that comb back. The liquid nitrogen will kill without physically disrupting the wax. And then you put those comb pieces back into the hive. A hygienic hive will recognize that everything is dead beneath that wax covering and clean it all out. And so in that way, it's hygienic. A non-hygienic hive will not clean it out. And that cleaning helps remove disease from the colony, not just the mites, but other bacterial diseases as well. One of the great things about this is we get to do lots of field work, and you meet all kinds of very interesting people, and sometimes capture funny pictures of bees doing something near you. The whole goal there was to find protein expression differences that related to the hygienic behavior. So we were finding things, this protein had a very positive correlation with hygienic behavior. This protein had a very negative correlation. That then gives us a handle to use a diagnostic to guide a selective breeding program, which is what we did. We then tested a traditional selective breeding program versus using a proteomic approach to selective breeding to select for hygienic behavior over time through 3 generations. So we started with F0 through F1, F2, and F3, measuring hygienic behavior with the conventional approach. You get a nice enrichment of the behavior. With the markers, we get an enrichment over time as well, not quite to the level of the traditional approach, but still reasonable. We then put that into a test where we gave bees these mites and the viruses that came along with them. Some of those bees were selected in the conventional approach, some were not selected, and some were selected using the proteomics approach. And then we let those bees go for a whole year, came back and asked, are they still alive, basically. And the unselected ones, these are either freshly imported or unselected Canadian stock, survived at 25 to 30%. Those that we selected using our markers, proteomic markers, survived at about 80%. And those that were selected by the conventional approach were around 70 to 75% survival. So the selective breeding works. This is the first demonstration of marker-assisted selective breeding in bees. And it's the first demonstration ever of the use of a protein panel for guiding a selective breeding program in any organism. Most selective breeding obviously would use a genetic marker rather than a protein expression marker. And we can get into why that might have worked if you want in questions. So moving right along, the other project I want to tell you about is the use of, or our ability using mass spectrometry to measure peptides presented on MHC molecules, both class 1 and class 2. So I won't get into the whole biology there. I'll presume that people know about antigen presentation. Maybe an incorrect assumption, and we can get into those questions as well. But basically, the MHC molecules present peptides from pathogens or from ourselves on the surface of cells, and those can then trigger a T cell response that will then lead to production of antibodies and killing of the pathogen, hopefully. And that's obviously the system that's taken advantage of in a vaccine. And so there's lots of interesting biology to do here, but the very practical application is in identifying a candidate antigen for a vaccine. And so the old way that, and it's sometimes still useful, way of developing a vaccine is just to take the pathogen, kill it or attenuate it in some way so that it doesn't cause a full-on disease, and inject that into a person or a cow or whatever, and allow their immune system to develop resistance to it. And that's how smallpox was first vaccinated against. There's reverse vaccinology, which is a popular approach in big pharma. Won't really get into it, but it's very, very, very laborious and not something that's really done in academia because A, it's boring, and B, we don't have the amount of resources that you'd need to really do it well. We're hoping to use mass spectrometry to identify candidate antigens from pathogens, and I'll talk about that. And then there's what happened with COVID and SARS-CoV-2. You basically guess. And I would say that the species known as Homo sapien got very lucky that the spike protein, it happens to be a very immunogenic protein, and we knew what the spike protein was and its role in coronaviruses. So it seemed to make a good candidate antigen, and it is the antigen in all the different vaccines, both our mRNA and other vaccines against COVID. But there was no real thought about, is there a better antigen? That was just the one thing that everybody went after. And it worked, obviously, for SARS, but it's not going to work for every virus or every bacteria or fungi that we might want to immunize against. So there's a field called immunopeptidomics, and there's different sort of versions of this name. But the idea being that you take some kind of cell that can present antigens, you challenge it with a pathogen. It might be a virus, might be an intracellular bacteria, might be an extracellular bacteria. The antigen processing machinery in those cells will process proteins from that pathogen, present them on the surface. Those are the things that are going to trigger a T cell response, and therefore those are the starting possible candidates for a vaccine. If we add acids to cells, then we can strip those peptides off of the surface, away from the MHC proteins that are presenting them on the surface. Then once we have those peptides in solution, we can analyze them by mass spectrometry and then do various things with them. So this is work that's been done by Queenie, who Alessia mentioned, and then more recently by Tisha and Lucy in the lab. We have applied it to a number of different pathogens to identify candidate antigens. So the first one we worked with was chlamydia. Then this is some data from MTB, tuberculosis, and we've also applied it to salmonella. But what I will say that we're doing now is going after this idea that most vaccines don't work in a large fraction of the world's population. And the reason, or one of the reasons, there are multiple, but one of the reasons is the diversity that we have in our MHC alleles in everybody's genome. Everybody in this room, unless your identical twin is sitting next to you, has a different set of alleles in your MHC genes. They're the most diverse of the genomically encoded genes in our body. And there's good reasons for this in terms of the evolution of different species being able to survive a pandemic. But everybody's got different alleles, and those have a big impact in the peptides that are eventually presented to the rest of the immune system. So we were able to obtain this series of B cell lines taken from a family, their mom and dad, a daughter and 4 sons, and then asked what antigens are presented on the surface of these cells. And so you don't need to be able to read very much from here. What is shown in each row on the left here, or on the right as well, is a different antigen, and then who it is presented by, grouped by the allele that they have in one particular MHC gene. So this isn't considering all of their genes, MHC genes, but just one particular one. And you can see that, and we're just using the reference numbers here rather than their names or anything else identifying. And so you can see these two people present a very different set of antigens on their cell surface, and those are different from this pair of people and these 3 other family members as well here. The point being that we have the ability to look at which antigens are presented by different alleles, and now we're scaling this up to look at a series of B cell lines from the 1,000 Genomes Project that attempts to cover the most of the diversity seen in MHC genes across the whole world's population, as a way to try to identify antigens or the characteristics of antigens that will actually be recognized and presented by as many people across the world as we can, so that we don't accidentally develop a vaccine against the next pandemic that only affects, for example, white Europeans or something like that. So I will end there. Thank again everybody in the lab. I didn't want to put up another picture because Armando had a great one in his talk. But as everybody who gives a talk, everybody like me who gives a talk knows, it's everybody in the lab who is responsible for all of the accomplishments. So thank you to all of them. And I also always end my talks with what inspires me, and that's outdoor adventures with my kids. So thank you for your attention, and the 3 of us will be happy to take any questions. You guys, come on up. Oh, I can't see who I'm looking at because of the lights, but I see a hand. So go ahead.
It's Peter. So great talks from everyone. I've got a question. Maybe Alessia is holding the microphone, so I'll start with you. Yeah. So that was a really interesting talk, and I think it's a great project. I'm wondering what is known about the timeline for the heart attacks that happen after hormonal therapy. And I'll follow up with why I'm asking this.
It should be on the green
Oh,
light side.
okay. So the timeline, this has not been very well studied in trans women, as that's an under-researched area in general. They have done quite a few large studies on the overall effects of hormone therapy. And the most recent one was done by Women's Health Initiative, and they had to cut it off at 10 years because they were seeing too many negative heart effects to be able to continue the study in like good ethics. So I would say by 10 years, you're definitely going to be seeing a lot more of that, but research on an exact timeline has not been done very well.
Okay, so if it's a matter of years, you might also want to consider endothelial cells or like cells that form arteries, right? And also cells that are important for lipid homeostasis, because if it's happening over years, it might be that not the cardiomyocytes are being affected, but actually your blood vessels are being affected. So those might be important to consider, okay?
Thank you for bringing that up. That is a very good point. The reason why we went with cardiomyocytes is because of the disease increase as well. I will look further into that. Thank you.
Okay. And then for Armando, I'm wondering how high throughput the lipidomics you guys can do is. Like, can you do a lot of different biological samples from different mice, for instance, and compare?
Yeah, basically for us, it's about the same work having one sample than 100 samples. Basically, after we acquire the data, then data processing. Lipidomics, I will say, is a little bit between proteomics and metabolomics in the way that we know how they behave a little bit more than metabolomics, so we can predict the retention time a little bit better. So that's allow us to trust more annotation of totally unknowns. Because lipids are very diverse, there are hundreds of thousands, you know, so there is impossible to have a standard for everyone, right? But we know how they fragmented and the length of the chain, we can see those fragments, so we can know how many carbons are, the double bonds. So the workflow that we have now, we can, depending on the sample, if we have a small amount, we like cells, a few cells, we see few of them, but if we have 1 microliter of plasma, for example, we can annotate 500 lipids easily. So yeah, we have this instrument that is the timsTOF. Basically, is the most capable instrument now in the market to do lipidomics, and we are very lucky that we have it.
Great, thank you.
Yep.
So Alessia, what sort of doses are they using in this transgender hormonal modification? The reason I bring this up was you mentioned 10 years for the nurses' health study. I wrote the original government monograph and got it wrong. I used case control studies. There were actually quite a lot of excess cardiac deaths in the first 2 years. Are the doses you're looking at in this hormone modification similar to that used in HRT? It's the same ingredients.
So to re-clarify, you're asking what the relative doses compared to what you would find in a cis woman, or?
No, I'm saying that we've got a huge amount of data on HRT, including from an excess of early cardiac deaths that were missed in the case control studies. So it's really a question partly of dosage. Would you be looking at, say, the numerous people receiving HRT for hormone replacement? Would you use that as a control to compare with your gender modification patients?
So you mean hormone replacement after menopause?
Yes.
Or in some other situation?
After menopause,
okay.
yes.
Yeah.
Okay. So the doses, I'm not too sure about the doses for the menopausal hormone therapy. I can say that the doses for gender-affirming hormone therapy do range depending on what method you're using and like the person yourself. Some people and also the doctors. Some doctors will start on a very low dose. Some will work their way up. Some might start with a higher dose as well. For blood concentration is how I'm going about this. I'm not sure if it's comparable comparing to the menopausal, but I will say it tends to be a little bit higher than what you would find in the average cis woman premenopause.
Thank you.
Yeah. Do you actually know what they would be relative to a relative cis woman premenopausal?
I might be able to answer that if you have.
No, I don't. I was hoping you're going to tell me.
Well, I've got a question. Leonard, I'm wondering if you take a peptide that may be produced by protein degradation and presented a purified peptide to an MHC-bearing cell, would that produce a different metabolic signature compared to a different peptide, with consequence to the immune response?
So you're meaning, like, does the process of going through antigen presentation have some impact on metabolism, and might that, or the metabolic profile, and might that change with what it's being? Yeah, it's a good question. I mean, at the very basic level, there's going to be a couple different amino acids depending on what gets in there, but I mean that's the obvious things. I would be shocked if there weren't broader differences, but how broad those differences are as a function of what is being degraded, I don't think I've ever come across anybody who's actually measured it, but we have the ability to do that now, so it'd be interesting. And
Alicia, you mentioned that you're going to be looking at male cells,
Yes.
male cardiomyocytes.
Yeah.
In my brain, I immediately thought, well, you need to be doing female cells as well to look at differences. Why would you not expect these hormones to be doing the same thing to female cardiomyocytes, and then you have zero difference?
So I think it's mostly cost why we're just going with the one cell line for now, but the reason why I'm expecting to see differences between the different sexes is because different sexes naturally have different amounts of estradiol receptors and different varieties present in their cell tissues. So the estradiol receptors are what's going to be triggering changes both in regulation of gene, proteins, etc. in the cell, and that's where I'm starting to think we'll see changes from.
You have done a fabulous job answering questions.
Thank you.
There's a lot of different combinations of things there to test, right? So at some point you have to just get ahead and do some work.
Yeah, and convincing her to do cardiolipids and some lipidomics or something.
Jason.
Armando, you manage a core facility to help people. And so I want to ask, if people want to work with you, do you have some basic advice on things that they can do to make their project most likely to succeed and make your life a bit easier?
Yeah, that's a very, very good question. So many times we run the samples and, they are coming from groups that are very well organized. So you basically trust the sample and you do all the data processing, and then when they ask you to do some statistics, and most of the time I'm okay working with samples that I don't know which group they belong. I think that's a good practice, that I don't have any bias at all. But then when I do the data processing, okay, tell me what is the control group, right? And they don't have a control group. So how do you compare? Then you waste a lot of resources, the time. So I think good samples and good experimental design is very key to save resources for everyone.
And if you haven't designed an experiment before, I'm just putting words into both your mouth and Jason's mouth. If you haven't designed an experiment before, consulting with the person who's running the facility is the best way to go to make sure you don't waste time or resources. Yep.
Okay, I don't know if the mic's working, but regarding the slide on immunopeptidomics and vaccine production using that in your lab, I might have missed this, but how does, or what's the function of the dendritic cells in the process?
Right, so I didn't really talk too much about that. the fundamental thing there is you want cells that are going to be able to present whatever antigens you are interested in. And so it depends what pathogen you're looking at. If you're looking at a virus, you can probably use almost any cell that the virus infects because there will be class 1 presentation on virtually all cells. We started getting into this area looking at intracellular bacterial pathogens, and most of the time there's very little presentation on class 1, so we needed professional antigen presenting cells. So that's basically macrophages and dendritic cells and B cells. So yeah, that's why those were mentioned there.
Thank you all very, very much for it.
Thank you. Armando, was this yours?